Hello everyone, welcome to Gebze Technical University Department of Bioengineering webinar series. This is our 28th webinar. Our guest for today's webinar is Dr. Burcu ERGÜN from Biotechnology Research Center of Ministry of Agriculture and Forestry, and the webinar title is Controlling Transcription in Non-Conventional Yeast, Komagataella phaffii, Promoter Engineering to Enhance Deregulate Expression. Let me introduce our guest shortly. Dr. Burcu ERGÜN received her bachelor's degree from Hacettepe University, Food Engineering in 2010, and she had also a minor degree from Chemical Engineering. She received her master's degree from Middle East Technical University, Department of Biotechnology in 2012, and she received her second MSc degree from University of Edinburgh, Department of Biotechnology in 2014. She continued her PhD study in Middle East Technical University, Department of Biotechnology. She worked as a visiting scientist at Burcu University, Vienna between 2017 and 2018 and completed her PhD study in 2018. Since 2014, she has been working as an engineer in Biotechnology Research Center of Ministry of Agriculture and Forestry, Ankara. Dr. ERGÜN, stage is yours.
Thank you for this introduction. Thank you for giving me this opportunity. I am very happy to meet with you all in this virtual meeting today. Today I will talk about promoter engineering in Komagataella phaffii to enhance a deregulate expression. Firstly, I will briefly mention about promoters. Then I will talk about some promoter design considerations, and at the end, I will give two promoter engineering examples. First will be on promoter engineering of ADH alcohol dehydrogenase 2 promoter to enhance recombinant protein expression over ethanol utilization pathway. I will mention about two different strategies in this context. First one will be modification of transcription factor binding sites. The second one will be nucleosome optimization. Then I will give another study on alcohol oxidase 1 promoter engineering to deregulate its expression. In this section, I will mention about targeted modification of transcription factor binding sites. In molecular biology, central dogma explains flow of genetic information in a cell. All the information is kept in nucleus within the DNA. Then this information transcribed to mRNA and translated to functional proteins. So gene expression is firstly controlled at transcription step, and promoter DNAs control transcription in a cell. What is promoter? Promoter is a DNA sequence. They can independently facilitate binding of transcription factors, then recruit transcription machinery on it, and initiate transcription of a coding sequence. They are used for a variety of applications in bioprocess engineering. In synthetic biology, to design genetic circuits, we need promoter elements. In metabolic engineering, to construct synthetic pathways, again, we need a variety of promoters. Finally, for recombinant protein production, it is one of the most essential genetic tools to determine production efficiency and productivity in a cell. So to accomplish all these applications, we need a promoter repertoire at varying expression strength and also regulated under different conditions, depending on our interested processes. Why we need to engineer promoters? Because it might be quite tedious to isolate and characterize endogenous promoters from organisms. Also, they usually function genetic context specific, and because of this, they have limited transcriptional window. And their most important drawback is they do not maximize the transcription levels achievable within an organism. So to overcome these limitations, we engineer promoters to modulate their transcriptional capacity and also to generate the necessary dynamic range to find gene expression to accomplish previously mentioned targets. Yeast promoter structure mainly composed from two different elements. The 5' element is called as upstream enhancer element. This 3' region called as core promoter region, core promoter element. On core promoter, RNA polymerase binds also this transcription apparatus assembled here. And core promoter determines transcriptional direction and start site. Coding sequence is located here after core promoter region. Upstream enhancer element determines transcriptional frequency and promoter strength. There are many transcription factor binding sites on it. Actually, there are transcription factor binding sites on core promoter region as well, but promoter strength is mainly determined by this element. When we talk about promoter architecture, we can talk about two different elements. Cis-acting DNA elements are DNA binding motifs for specific proteins, regulatory proteins. They are DNA sequences, those cis-acting elements. Trans-acting regulatory factors are transcription factors or other regulatory proteins that are specifically bound to their known motifs. And together with cis-acting elements, trans-acting factors, and bound other proteins and three-dimensional structure of promoter compose promoter architecture. There are different promoter engineering approaches. Random mutagenesis randomly mutates DNA by error from PCR. They usually create a wide range of promoter library, but they have a drawback. Since they randomly mutate the promoter DNA, they usually disturbs functional transcription factor binding sites, so their library is usually weaker than the parent promoter. In saturation mutagenesis, they specifically mutate spacer DNA regions between functional motifs, functional transcription factor binding sites. In chimeric promoter design, they are evaluating these two elements as a separate engineering tools. They combine different core promoters with different upstream enhancer or upstream activation sequences. They can also tandemly ligate different upstream activation sequences with a core promoter to increase the strength of the promoter. In Yarovia lipolytica, to my knowledge, the strongest promoter was obtained with this chimeric promoter design strategy. Synthetic promoter still relies on native cis-acting DNA elements, but they combine them in an unconventional way. They combine different cis-acting elements to create synthetic promoters. But as you can see, all promoter engineering approach mainly targets transcription factor binding sites. In this presentation, I will talk about how to rationally design a promoter by targeted modification of a transcription factor binding site. Before designing a promoter, we need to think about many parameters and many constraints. Firstly, we need to know native transcription factor binding sites found in the natural promoter. We can identify them either by bioinformatics tools or by using publicly available databases. We can manually create them. Throughout this putative TFBS identification process, we need to consider both strands because transcription factor binding sites can function bidirectionally. So putative cis-acting elements found on both strands are important for us. Also, when we change the distance between cis-acting elements, their corresponding transcription factor binding sites interaction can be changed because they can come closer, they can go further. So distance between these native functional cis-acting elements should be preserved and kept constant. Also, when we change the promoter length, it's probably three-dimensional structure will change and will affect many things in the promoter. To keep this length constant, we can introduce our modifications by point mutations or replacement of a gene sequence with a targeted synthetic DNA element. Another important issue is distance from the transcription apparatus. When our engineering sites come further from the core sequence, the influence of that introduced transcription factor binding sites get less. So it's better to make modifications in close proximity to the core sequence. Since both strands can harbor DNA binding motifs, and also sometimes some motifs are overlapped within a DNA. They can share some nucleotides, or they can share totally almost all region. So when we introduce a mutation to a transcription factor binding sites, we might remove overlapping motifs, or we might generate non-design sites. At the end of our design, we need to keep this issue in our mind. We might see some unintended effects at the end. In eukaryotic promoters, nucleosome structure is quite important because it has a blocking effect for TF binding to DNA. So while considering our engineering targets within a promoter, we can think about nucleosome depleted regions, or we can introduce nucleosome disfavoring poly-DA-DT tracts at the 5' and 3' ends of our engineering cis-acting sequence. Certain TFs function cooperatively within a cell. For example, in Saccharomyces cerevisiae, ADR1 and CAT8 collaboratively activates many genes, many gluconeogenesis genes in Saccharomyces cerevisiae. So if we think about introducing a cis-DNA, cis binding site for one of them, we need to consider to integrate second point for the second one, because they may not function alone without their cooperative factor. Also, these engineering sites should locate in close proximity with appropriate layout design to allow them interact with each other and cooperate to the transcription activation process. Also, some transcription factors function as a homodimer. Again, for example, Saccharomyces cerevisiae ADR1 binds as a homodimer and activates transcription. So for those factors, we can introduce a palindromic binding motif with a proper spacer DNA element. I will give two examples. The first one will be engineering of alcohol dehydrogenase 2 promoter architecture to enhance recombinant protein expression on ethanol. The second one will be engineering of alcohol oxidase 1 promoter for deregulated expression. This study was published in Biotechnology and Bioengineering in 2019. In this study, firstly, we determine our native ADH2 promoter architecture. For this purpose, we firstly analyzed our ADH2 promoter by a bioinformatics tool, MetInspector. So we obtained many putative transcription factor binding sites. Also, we used yeast-tract TFBS database, and by using information from it, we manually created our promoter to find putative TFBSs. Also, we considered nucleosome positioning. For this purpose, we analyzed the nucleosome structure of ADH2 promoter by NUPOP, Nucleosome Position Prediction Tool. And this is refined structure of the ADH2 promoter, and many more elements were found initially. There are two nucleosome depleted regions. One of them is here, the other one is here. And throughout our manual creation, we find 3 MXR1 binding palindromic repeats. And also there is a single one here. Also, since we are targeting ethanol utilization pathway, we consider 2 more transcription factors. The first one is CAT8 TFBS and CAT8 transcription factor. The other one is ACA2. I will only mention about our successful variants in this part. We engineered the ADH2 promoters by introducing synthetic CAT8 binding motifs. In our first design, we chose an engineering location 16 base pair upstream of the MXR1 binding region, because from the Saccharomyces literature we know they are functioning together and collaboratively. When we did this design, we used EGFP as a reporter protein. In ethanol fermentation, we see 2.4-fold stronger expression potential by this promoter. And we decided to design another promoter variant, again with cat8 synthetic binding site. In this case, we bring our site closer to the coding sequence, so we expect its influence will be higher. Also, in this region, there are two neighbor MXR1 binding palindromic repeats. So they can, these two can function together. Also, these two can function together. It can be better in terms of their collaboration and transcription activation. Our expectation was quite true. At the end, we obtained 4.8-fold stronger promoter variant with this design. In our second part, we decided to optimize nucleosome structure. This figure schematizes the nucleosome structure briefly. When his DNA is wrapped around histone proteins, there are these TFBSs here, but those TFs cannot come and interact with them because histone doesn't let them come and interact. When we unwrap the DNA, these binding motifs become free and they can easily interact with their corresponding TFs and start transcription activation. So our aim was making our promoters nucleosome affinity lesser to make them more available for TF binding. Throughout nucleosome optimization, we protected some regions. Those regions are the core promoter region, yeast stress responsive elements, carbon source responsive elements, non-optimal carbon source utilization related transcription factor binding sites, and originally nucleosome depleted regions, which we thought they can harbor some important regulatory elements within those regions. So we protected those regions from any mutation. Also, we provide transcription factor binding site database obtained from yeast tract to the algorithm, and we prevent the deletion or insertion of any known TFBS. So we wanted to see only the nucleosome optimization effect in the promoter strength. This is the schematic representation of our nucleosome optimization. We performed 100 rounds of optimization. So at the end, 100 nucleotide was mutated to obtain PADH2 knockout, and the uppest one is wild type ADH2 promoter. This lowest one is the PADH2 knockout promoter's nucleosome affinity structure. When we performed this, our promoter variant upregulated expression on ethanol 1.5-fold. On methanol, 3.2-fold. So it was quite promising engineering strategy for Pichia Pasteur's ADH2 promoter. At the end, we decided to combine two superior tools. These are nucleosome optimization and cat8L2 modification. But when we did this, we had some unexpected results for ethanol condition. We didn't see additive effect of two engineering tools. Our promoter showed 3.7-fold stronger expression level on ethanol compared to wild type promoter. This strength is between of the both parent engineered promoter strength. But on methanol, those two modifications show synergistic effect. We obtained 8.8-fold increased expression potential. I also didn't mention, we constructed many other promoter variants by using ADR1 cis-acting elements and ACA2 cis-acting element, but they didn't have neither positive nor negative effects on promoter strength and regulation, so I didn't mention them. Also, we introduced a Tata binding domain, Tata box, into the core promoter. In this case, we obtained weaker promoter variant. Its strength was reduced around 30%. To confirm, to evaluate the strength of our strongest promoter variant, we evaluated its expression potential with Komagataella phaffii benchmark promoters. AOX1 is here. Actually, as you can see, compared to AOX1 expression potential on methanol, ADH2-cat8L2 promoter shows more than 2 times higher expression potential, but it's produce recombinant proteins on non-toxic carbon source, ethanol. It has another advantage. We need to confirm our promoter strength with a second model protein, because in some cases these results can be obtained by proteins specific to the reporter protein. We used human serum albumin as a second model protein. We obtained similar results to the EGFP expression levels. With nucleosome optimized variant, we obtained 1.4-fold higher expression level. With ADH2-cat8L2 variant, we obtained 4.3-fold higher product yield. I'm going through my second study now. Komagataella phaffii, actually formerly known as Pichia pastoris, it's methylotrophic, non-conventional yeast. Due to its methylotrophic nature, most of its strong and regulated promoters are obtained from methanol utilization pathway. Alcohol oxidase promoter is one of them. These promoters are strong and tightly regulated, but they have a drawback. They are induced mainly by methanol. Since methanol is toxic, and in food industry, it's usually crude proteins, crude enzymes are used. So methanol contamination is not wanted. Also for pharmaceutical protein production, during regulations and approval of a protein, again, methanol usage as a inducer can be problematic and can make this regulatory step harder. So we decided to change the regulation mechanism of alcohol oxidase 1 promoter. This represents the native regulation mechanism of AOX1 promoter. It's strongly induced by methanol and repressed by ethanol, excess glycerol, and excess glucose. We decided to mimic the architectures from the nature. And the most information was obtained with Saccharomyces cerevisiae. For many decades, its transcription mechanism has been investigated, and more information is accumulated there compared to Komagataella phaffii information. So we decided to use Saccharomyces cerevisiae ADH2 architecture as a model. It is mainly activated by ADR1 and cat8 transcription factors. Also, we realized another transcription factor, ACA2. ACA2 null mutants showed growth defect on ethanol in Saccharomyces cerevisiae, and they defined ACA2 as a stress-responsive transcription factor, and it's involved in utilization of non-optimal carbon sources. So it seems as a promising target for us. Also, it's important to engineering locations because, as I mentioned earlier, many parameters affect availability of those cis-acting elements. MXR1, methanol expression regulator 1 transcription factor, is the master regulator of alcohol oxidase 1 promoter in Pichia pastoris. And 6 MXR1 response elements was identified on alcohol oxidase 1 promoter in the literature. So we decided to use those sequences as our engineering targets. In our first engineering study, we changed this MXR1 binding motifs with Saccharomyces cerevisiae ADR1 palindromic repeats. The logic was this here. Actually, ADR1 and MXR1 are homologs. In Pichia pastoris, ADR1 homolog is called as MXR1. In this case, it is like this. We introduced 2 different MXR1 binding sites instead of 1. Also, we reduced the spacer region between them. Because Saccharomyces cerevisiae ADR1 binding affinity to its binding motif is higher compared to Pichia pastoris MXR1 binding affinity to its MXR1 recognition element. So we aim to allure more MXR1 proteins here, so better activation could be achieved. This was the idea. But at the end, it didn't change regulation at all. We didn't see any positive or negative effects in this engineered variants. In our second strategy, we introduced cat8 binding motifs by replacing MXR1 binding motifs. In the first design, AOX1 cat8 L1 design, we didn't see any difference. It behaved like a native wild-type AOX1 promoter. But in the second design, when we introduced cat8 instead of MXRE3 region, 1.8-fold stronger expression was achieved on methanol. This was actually a kind of unexpected, not unexpected, surprising result for us, because in the native structure of AOX1 promoter, there wasn't any cat8 binding motifs, and so we didn't expect any positive regulatory effect of it in methanol metabolism. But they function quite well. When we introduced cat8 binding motif to this third site, again, it increased the expression strength on methanol 1.7-fold, but we had very good, nice results in this case. Ethanol repressible AOX1 promoter is converted to ethanol inducible PAOX1 cat8 L3 variant. On ethanol, it showed 70% of expression level of methanol-induced AOX1 level. So with a single TFBS modification, we achieved to change its regulation mechanism on ethanol. In our third synthetic binding site, we introduced a ACA2 binding motif here. This is again surprising. We expect to see some improvement on ethanol, but it enhances the expression strength of AOX1 on methanol. Around 40% higher expression was achieved with this variant. After this, we decided to combinatorially apply those engineerings to the AOX1 promoter. We see the most influential effect with this cat8 L3 modification. We didn't see any influence with this ADR1 modification, but we wanted to see their combinatorial effect in Pichia pastoris AOX1 promoter. When we combined them, its expression level on ethanol increased around 10%. But on methanol, some reduction occurred. When we include ACA2 binding motif together with this cat8 and ADR1 modification sites, in this case, there was an increase on methanol. Actually, it was quite obvious. ACA2 contributes to methanol activation in Komagataella phaffii. Again, on ethanol, some reduction occurred on the expression potential. So we wanted to make a strong ethanol-regulated AOX1 promoter. We decided to apply all three ADR1 modifications together with all three cat8 modifications, TFBS modifications. When we did this, we created the strongest ethanol-regulated variant. It becomes stronger than wild-type AOX1 on ethanol. On ethanol, it produced 1.3-fold higher recombinant protein production compared to wild-type AOX1 did on methanol. Also, on methanol, it showed quite a higher expression. It was around 80%. It was producing around 80% higher recombinant protein compared to wild-type AOX1 promoter. We also wanted to introduce ACA2 cis-acting element to this structure. And this is our strongest variant, PMAOX1. On methanol, it shows almost 2 times higher expression potential than wild-type AOX1 promoter. Again, on ethanol, it's quite strong. It produced 1.3-fold higher recombinant protein compared to wild-type AOX1 did on methanol. This is our promoter library. We designed different promoter variants. They can show activity on both ethanol and methanol. They are still, all of them are still repressible on glycerol and glucose. So we kept a tightly regulated mechanism of AOX1 while making them ethanol responsive. This is again second model protein study with our most promising variant AOX1. As you can see, productivity on this is wild type AOX1. This is modified AOX1. Productivity on methanol of AOX1 and ethanol of MAOX1 is quite similar, but cell yield on ethanol is quite high compared to cell yield on methanol. So product yield per gram cell weight reached around 55% of wild type AOX1 on ethanol. But on methanol, it produced 1.58-fold higher recombinant human serum albumin compared to wild type AOX1. We recently wrote two methods about how to design engineered promoters. These are prepared for methods in enzymology series. If you're interested in promoter engineering, maybe they can be useful to have a look. Thank you so much for listening. If you have any questions, comments, I would like to hear.
Thank you very much for your nice presentation. If you have any questions, we can have right now. Any questions?
Merhaba, Barış Hoca biraz önce buradaydı ve ben onun da çalışma alanıyla birazcık kesişme var. Belki onun yorumları daha şey olur diye düşünmüştüm. Teşekkür ederiz her şeyden önce. Yani promoterların engineer edilmesiyle productivity'nin arttırılması neredeyse her proseste şart olan bir şey. Yani ekonomik feasibility bakımından. O yüzden sürekli çözümü aranan bir problem ve kombinasyonlar sonsuz. Elinize sağlık.
Sorusu olan yoksa webinarımızı burada bitirebiliriz. Çok teşekkür ederiz tekrardan katılımınız, güzel sunumunuz için.
Ben teşekkür ederim.
Teşekkürler tekrar.
Önümüzdeki webinarlarda görüşmek üzere. Hoşça kalın.
Hoşça kalın.